לא. אז אני לא הולך. אני לא הולך לעבודה כי היום נהרס גם ככה. שזה גם נוח. מה זה?
כן, שוב הוא... שאם הוא בא בלילה והיו פחות מ6 שעות שינה, טוב לא יצא מזה.
מישהו מכיר את המערכת כאן? לא יודע, יותר מבין? שאני לא מכיר את הכל כך טוב. כאילו אין סאונד, אני מנסה להקרין את זה, ואין סאונד. שיש רגע. טוב. טוב. אז so we'll start with answer together the question page for transport of ions. Okay, let's start with the first one. So, first question. Below are 3 steps from the operational mechanism of a protein that transfer an ion from one side of the plasma membrane to another. Okay, so that you have some principles or basic steps in the mechanism of some transport protein. So 1, ion binding. 2, conformational change. 3, release of ion. These 3 principles to which transport protein do they hold? To which are they true? So they can be found among additional ones in the operational mechanisms of voltage-independent ion channels. So what do you think? So the voltage-independent are the voltage non-gated, the ones that don't have no gate, no domain that serves as a gate. So what do you think about conformational change in these proteins? It's not too obvious that they have any. So this is not true for these proteins. ATP-driven or ATP-powered pumps, yes, that's true. Symporters and antiporters. Yeah, so, well, both of them have sort of the same principle, only the one delivers for 2 opposite sides and so on. So yes, C and D is also true. So we have an E is voltage-dependent ion channels. What do you think? Because again, it's a channel, and if you say that there is there's no ion binding group, not right. That's the most accurate thing to say. So there's no ion binding. So it's only true for B, C, and D, which is H. Okay. Number 2, in what aspects do different ion channels differ between them? In the type of the transferred ion, yep. In the transfer rate, yes, we've mentioned this. Dependency or independency on the membrane potential. So some of them are sensitive to the membrane potentials, while others are not. And D, dependency or independency on the ligand, on a ligand. So again, to say that there are sensitive to some stimulus, to some. So H, all answers are correct. 3. During the transfer of the potassium ion of potassium ions across the potassium channel, the ion is surrounded by water molecules. Right? Remember, it shield off its water shell and passes sort of naked without these molecules. Okay, so this is incorrect. B, the ion shed its water molecule envelope, exactly what you just said. C, the ion interact with other ions that pass in the channel. And let's continue with D for a second. And so D, the ion interact with other ions of the channel. Yes, D. So B and D. So we described the principle that really the ions leave their space around them to one another, so they pass in spots 1, 3, and 2, 4, not to collide with each other. So it's exactly what C is based upon. 4, in the operational mechanism of the voltage-dependent sodium channel, the refractory period is achieved due to, so A, conformational change induced by a movement of the potential sensor. So we're talking about the refractory period specifically. So conformational change induced by a movement of this is the step that opens the channel, but it's not the one that prolongs its opening once it is opened. So I would say that A is incorrect. B, membrane potential cannot go over a certain threshold. No. Even if it is true, then it will not serve as any delayed aspect in the mechanism of this channel. And C, the protein globular region binds the channel gate and shut it off or close any further ion current through this channel. So it's C. Only C. Okay, last question. Which of the following is a mechanism of termination of synaptic signal? So we have E, diffusion of a neurotransmitter from the synaptic cleft. Yeah, just passive diffusion, so that's one of the mechanisms. B, reuptake of the neurotransmitter back to the presynaptic site. Again, something that will clear it from the synaptic cleft and will cause termination. C, phosphorylation of the neurotransmitter on the synaptic cleft. No, just no. D, hydrolysis of the neurotransmitter in the synaptic cleft. For some neurotransmitters, it is the mechanism. So we have ABD, which is G. Okay. By the way, how do you feel with the question pages so far? What is your rate of success? Well, are the questions clear in the manner that they are phrased?
It's really clear, but not always. You don't always find the answers in the
You don't also find maybe the specific answer. That's okay. If the answer is between the lines, that's still fair. What do you think? Really didn't talk about. Okay.
Even the diffusion of the neurotransmitter from the synaptic cleft.
Yes.
It's about reuptake and hydrolysis. That's not diffusion.
Okay. I did mention it verbally. I didn't write it maybe on the slide, and maybe I should have, and I can fix it for next times. But some material passes. Not all 99% or 90% of the material would be in the slides, but maybe not all of it. Not because it's a trick or to trick anyone, just because that's the way it goes. But I can add it as a clear additional factor that affect signal termination. Okay, what do you want to say?
The last question sheet, that there was a question that
The last sheet, yeah.
there were a few terms that we didn't learn.
Few times. Well, there was the time when I asked about microRNAs and siRNAs, and we still didn't learn it. Okay, so that was absolutely my mistake because of some changes we did. Well, this is נו, יוצא דופן.
Exception.
An exception, sorry. I like to think it's an exception. Okay, but again, this is just the question pages. I like to believe serve as something to give you an intuition of what you really need to know at the end of the course. That's the only way I can answer this question, which is always being asked. Okay, so let's proceed with the next chapter. G protein couple receptors. It's parallel to the textbook to chapter 13. So remember we talked about the general discussion about transport of ions and other molecules across cell membranes, and we mentioned at a certain point a family of proteins that that are not, or a family of receptors that are not a channel as they are or by their own, but they affect another protein, and most commonly they affect a G protein. So these receptors are called G protein coupled receptors because they are activated by, or they activate the G protein, and so they are coupled to it. And these receptors, well, they found in all eukaryotic cells in nature. There are several hundreds of genes that code for them in the human genome alone. There is an estimation I read once, which is really kind of, it's backed on good and sound evidence, that it's about 50% of the approved drugs today on the market. Well, you know, for most of the drugs, we don't have a mechanism of action. We don't know how they work. We just know they work empirically. And for about half of them, we think that the mechanism of action passes through G protein coupled receptors. Maybe just one more word of motivation about these proteins. So they function in so many bodily functions and senses. They function in vision, in smell, in taste, I think in touch also, in digestion, in neuronal plasticity, and many other mechanisms. Anyone knows which is the sense that has the biggest array of receptors? Smell, right? Where you have a receptor for each and every odorant, each and every chemical molecule at the outside world, at the odorant space. So it has, Amidim Mizrahi will talk to you about that in physiology course next semester, but we have more than 1,000, I think, more than 1,500 even, receptors that are coded for the sensory smell alone. Sensory smell for the smell alone. So, okay, so these are very important proteins. That was the first message to deliver. And now some word of overview, even before the overview itself. So there's an enormous variety of molecules, signaling molecules that can be found in our body. Some of them that function in cell-to-cell communication. They can be amino acids, that we've already met some of them, lipid derivatives, small peptides, and even not so small peptides, even proteins. And all signaling molecules act as a ligand that is recognized by a receptor via a combination of weak interactions, and which then activates the receptor in some manner. And the whole process of the whole process from the binding of the signaling molecule to the receptor through activation of some molecular cascade and ending with some intracellular process that have initiated is called signal transduction. And this will be the focus of today's chapter. So the whole process is termed signal transduction. And a general look of molecules that play a role in cell-to-cell communication show that we have several different strategies to activate intracellular processes. Some receptors activate kinases that phosphorylate other proteins, transcription factors, that are then translocated to the nucleus and induce transcription of new genes, synthesis of new proteins. Some of them phosphorylate directly transcription factors, again, that translocate to the nucleus and induce transcription. Others change the conformational structure of a protein that again changes its function, and so on and so forth. And you see lots of different strategies. And G proteins act through all of them. And we'll see some specific examples later on. Now, all of these examples are for molecules, for ligands, that are recognized by a receptor, by a membranal receptor, a receptor that sits on the plasma membrane, and they interact with it from the exoplasmic side, from outside of the cell, which makes sense. But just to complete the picture, let's just mention that there are some molecules that have the ability to penetrate the membrane, and these molecules, signaling molecules, would have receptors inside the cytosol, and the biggest family of such molecules are steroids. A molecule is called steroids, and you can read a little bit more about them at home. So they would have receptors that are cytosolic and not transmembranal. Okay, so these are molecules that play a role in cell-to-cell communication, and this will really be the focus of today's class. But you know, there are some molecules, again, just to complete the picture, there are some molecules that play a part in communication between different organisms in nature. Do you know what molecules I'm talking about? So there is some chemical molecule that transmit messages, not between cells inside your body, but between different organisms in nature. Pheromones, right. So again, I think Adi Mizrahi will mention some principles of function about these molecules. There is some interesting evidence that there is pheromones in higher primates, including humans, but it's kind of a gray area still.
They are G protons as well?
Well, I'm not sure about the mechanism of function. I just wanted to mention them again as a contrast to, we are talking about cell-to-cell communication today, and there is also outside of the body communication between organisms in nature. I really don't know about the mechanism. In mice, I do know that some of them pass through G protein coupled receptors. That's all I can say. Okay, so we will start with a general overview of intracellular signal transduction, some principles. Then we will meet the G protein family, some principles of structure and function, and then we will focus on three distinct pathways through which G protein act. Those that act through the pathway of adenylyl cyclase, those that regulate ion channels, those that act through phospholipase C, another major player in cells, and we will end with just making some order in the principle that we will collect along these points. In the last point, activation of gene transcription by G protein coupled receptors, just to again emphasize the principles of the mechanism of action of gene transcription that is induced by G protein coupled receptors. G protein coupled receptors are sometimes called GPCR, so I might say GPCR every once in a while. It means the same thing. Okay, so signaling molecules in our body are classified to three classes by the distance that passes between the cell that has secreted them to the cell that receives them. So endocrine signaling is the signaling of a molecule between faraway cells. This is where the distance between the cell that secreted the molecule and the cell that receives it is the biggest. And for instance, it can be an example of endocrine signaling is a molecule that's secreted in some place in the brainstem, say, and that affects the sexual development. So it affects our sexual organs in a faraway site. So these molecules have to be carried by some medium, by some substance, and in the body it is most usually the blood. And what other fluid? What's the fluid that inside our brain? Cerebral spinal fluid, which also carry hormones or other endocrine molecules. So what's the cerebral spinal fluid is built of? What does it include? a lot of water, but not just water. So because we mentioned it, so so it has proteins in it, low levels of proteins. It has glucose, because it's a nice, you can deliver glucose through it. It has white and red blood cells in it. And now you know that it also carries hormones. Okay, in paracrine signaling, the signaling molecules affect the close vicinity or affect a cell in the close proximity of the cell that has secreted the molecule. So the most common evidence example that we've already described so many times is the neurotransmitter at the synapse. Synapses are example most of the time for paracrine signaling. A cell that affects a nearby cell. Signal to a nearby cell. And we also have a weird process of autocrine signaling in which a cell secretes a signaling molecule and then internalize it back again to read the message that it itself wrote. So cells write messages to themselves. Kind of sad, don't you think? So it's a weird process because why bother go through all this mechanism of secrete a vesicle and then take it back again and so on? And as we mentioned in previous times, we think that this process is evolutionary. Along the evolutionary development have been based or used mechanisms that have already been existed through these mechanisms. And so we sometimes see, very inefficient processes in nature, and we believe it's because of a relatively late evolutionary process. Okay, so that's autocrine signaling. And as we mentioned very laconically last time, some signaling molecules can act in more than one way, in more than one signaling strategy or signaling system. They can act both short-term or long-term. And an example that was mentioned here, epinephrine is a neurotransmitter and a hormone. It acts both in paracrine signaling and in autocrine and in endocrine signaling. Okay. So when a cell receives a signal, it will most usually change either the function of a pre-existing protein. It will change the status of the function of some protein that is already there at the cytosol, or it will change the quantity of the protein. So either way, we change the final outcome, the substrate on which the protein acts upon, either through changing its function or changing its amount. And again, G protein coupled receptors act on both ways. Okay, now there is some divergence when we move from the level of receptors to the level of intracellular signaling that are activated downstream. Because some classes of receptor can initiate signaling via more than one intracellular pathway. They can activate more than one intracellular pathways. So there is some divergence between this level, the level of receptors. If these are the receptors on the plasma membrane, and these are intracellular processes, like a very small neuronal network, there is some divergence when we move from this level to this level, because some of these nodes can affect several downstream nodes. But there is also a convergence when moving between these two steps, because despite the huge number of different kinds of receptors, there is relatively restricted number of intracellular processes that can be activated. So, for instance, highering the levels of inside the cell cyclic AMP is a very common, very frequent end step in a long cascade maybe of activation upstream. So activation of various receptors can converge on this single intracellular process, and so we have also diconvergence. No, other color. Okay. Right, so each receptor at the plasma membrane recognizes and binds to a single molecule or to a very small family of very similar molecules having a really almost the same domain of binding. So each receptor binds only a single signaling molecule or a very small bunch of very similar molecules because of its structure. In contrast, a single signaling molecule can be recognized by several different receptors. So the first principle is manifested in experiments where you mutate, you perform point mutations for instance, a growth hormone, which is a signaling molecule, and point mutations at the corresponding receptor. And the pink amino acids here and the blue and yellow amino acids here are the ones that if you change them, you affect very robustly the binding between the signaling molecule and the receptor. So these amino acids marked in pink, yellow, and blue are essential for correct and stabilized interaction between these two molecules. Yeah, of course. So it's enough to change the signaling molecule just a bit in one or just several amino acids for the receptor not to recognize it at all. And so as we said, each receptor binds only a single signaling molecule or a very specific domain in it. And the second principle, which is again sort of a principle of divergence, is manifested, for instance, in the example of acetylcholine. So this signaling molecule is a specific signaling molecule, and it can act on several tissues in our body through several different receptors, and each time induce a different effect. So acetylcholine, if it will be recognized by ligand-gated ion channels on muscle cells, it will induce contraction of the muscle. The same signaling molecule, if it will be recognized by heart cells, they will cause via a G protein coupled receptor a slowing of the heart rate. And the same molecule, if it will be recognized by pancreatic cells, it will cause secretion of digestive enzymes. Okay, second messengers are intracellular, relatively small molecules that function in the direct activation of intracellular signaling. They are called secondary. Why are they called secondary messengers? To who are they secondary? Who's the first messenger? Not the receptor, but not the GL, but the ligand. Yes, the neurotransmitter or the signaling molecule from outside the cell are the first messengers, but no one calls them this way. But then inside the cell, you have the secondary messengers, which continue to transmit the signal. So most commonly, secondary messengers are cyclic nucleotides with a single phosphate group. Here you see the cyclic adenosine monophosphate and the cyclic guanine monophosphate. Should I say guanosine? I'm not sure. The cyclic GMP. Secondary messengers can also be derivatives of fatty acid chains. And this is also a very common example, some derivative of a sugar molecule. So these are the most common secondary messengers found in cells. And again, if that's the input that you insert the cell, it's not enough to say, as in the case of action potentials that flow into a neuron, it's not enough to ask what is the input, but you have also to take into account what's the state of the neuron, or in our case, the cell, what's its state while it was introduced with an input to understand the output. So the same molecule, the same secondary messenger in different cells will cause different outcomes simply because of different microenvironment around and inside each cell type. So again, in muscle, okay, so by the way, secondary messengers can also be just ions, as we saw in the Candel paper. There it was the calcium. So a rise in cytosolic calcium in muscle cells will trigger contraction. In endocrinic cell will induce exocytosis of secretory vesicles, and in neurons it will induce exocytosis of neurotransmitters. So again, the input is not enough. You also have to know the state of the neuron or the state of the cell, what mechanisms are available in it at any given time, not only cell type-wise, but also along the development of a neuron or along the development of any other kind of cell. Its state changes, and different states will cause different outcomes given the same input. And that's partly why we still don't know to predict not only how neurons will behave upon any stimulation, but also we even don't know to predict how pancreatic cell will behave upon exposure of some hormone. Not really, not accurately enough to deal with it or yeah. Sort of, yes. Okay. So these were secondary messengers. Yes.
So the calcium itself is not a secondary messenger.
Well, people do call it sometimes a secondary messenger. The calcium ion itself. It can be due to influx of calcium from outside the cell. Still, no problem, terminology-wise. Still, people call it a secondary messenger. That's the most, precise definition that people give to secondary messengers. They are just small molecules that transmit the signal inside the cell. Activation of almost all cellular or membranal proteins go through, virtually all of them, as much as I know, go through the process of adding phosphate groups or removing phosphate groups from other intracellular proteins. So let's mention the proteins that that add phosphate groups. As you remember, these are called kinases. And the proteins that remove phosphate groups from inside the cell, proteins which are called phosphatases. And just one more word about them. So there are two types of protein kinases. There is the class that add a phosphate on the hydroxyl of tyrosine, and another class that add a phosphate group on the hydroxyl of serine or threonine. They have distinct function and structure. Now, I think we have mentioned it before, but each protein kinase phosphorylates a specific sequence, specific structure on the target protein. So it's a function of structure and sequence on the target protein. And each protein inside the cell is most usually subjected to phosphorylation by more than one kinase. More than one kinase. Okay, some components of the signal transduction machinery are clustered spatially at the same site. So this picture is a very typical one for the postsynaptic site in synapses. And in postsynaptic site in synapses, you will most usually find an adaptor protein. So an adaptor protein is how we call a protein that links between two additional proteins, and sometimes even more than two. So it's an adaptor. And it's a very famous adaptor protein, PSD95. And it has several domains in it, several motifs, and the most characterized one is the PDZ domain. It's a 90 amino acid long domain that captures 3-letter or 3 amino acid sequences on the target protein. So PDZ will recognize some consensus motif on this protein and on this protein and on this protein, and the consensus sequence will be the following. Well, it has sort of 2 consensus sequences. It can be just that you get the feeling of what the consensus sequence in proteins mean. So it can be either serine or threonine, any amino acid, any hydrophobic amino acid, quite degenerate, you must say, quite general, or it can even be more general, hydrophobic amino acid, any amino acid, hydrophobic amino acid. So we really don't think it's the only determinant that determines the interaction between PDZ and other proteins, but we do know it's a consensus. Yes, Jonathan?
Serine and threonine are both hydrophobic?
Yes. Serine and threonine are both hydrophobic. But still, I'm not sure how did they get to these different, because then it's a specific case on second consensus. I'm not sure. Okay, so PDZ, for instance, will interact with the NMDA receptor at the postsynaptic sites. It will interact with some adhesion protein that's called neuroligin, and with other proteins, for instance, other glutamate receptors. And through additional adaptor proteins, it will find it to eventually even interact with the intracellular cytoskeleton. Yeah, for instance, the actin component of the cytoskeleton. So this whole cluster of proteins localize all the proteins that need to be localized or need to be nearby spatially at the same point in space due to these adaptor proteins. And the most common one, which we have described, is the PSD95 with its PDZ domain.
So the PSD95 only works with the PDZ?
What do you mean by only works? Well, it has the PDZ domain in it is the one that links to other proteins and allow its adaptor feature. Allow it to adapt between different proteins, between additional proteins. The PDZ is a domain inside the PSD95. PSD95 is the whole protein. Yeah, that's PSD95. And inside it has these binding domains and other binding domains. But the most common one is PDZ.
Okay, so
okay?
But you said it's an adapter, so it binds between
a NMDA glutamate receptor and other glutamate receptors and other proteins and some adhesion molecule that stick the whole thing to the plasma membrane and so on.
So it keeps a special.
Right. It keeps all these components together at the same location in space. Okay, so the signal termination, we've discussed signal termination at the synapse site, but here in signal transduction that goes through G protein coupled receptors, you can have signal termination by degradation of a second messenger. That will terminate the signal. Deactivation of any component in the signal transduction pathway, but not that of the receptor. Any deactivation of the receptor happens only after a prolonged, robust stimulation, what we call a tetanic stimulation, and only then we move to another area, another domain of regulation of signal termination that affect the receptor. So again, the common signal termination happens either on the second messengers or through a deactivation of any component in the signal transduction pathway other than the receptor itself, the membranal receptor. And if you will stimulate the cell strong enough, you will expose it to the ligand, to a hormone, for instance, long enough, then other things will happen on the receptor. So, for instance, we can have internalization of the receptor, just removal of it from the plasma membrane, or change it conformationally to give some conformation in strands that cannot bind a ligand, or change it conformationally so it binds a ligand but cannot induce the downstream effect. It cannot activate the G protein, for instance. Yes.
You said that the activation of transduction of protein, which is not receptor, and if you stimulate it long enough, this is the deactivation of the receptor?
Yes. Yeah, these three are the deactivation of the receptor that happens after a prolonged stimulation. Exactly. And the first two happen all the time. Okay. So these were some principles about the intracellular signal transduction, and we'll see some specific examples of them later on when we will discuss specific pathways. Let's take 15 minutes, not more than 15. Let's come back a few minutes before 9, and we'll proceed with overview of G protein structure and function.
can I disturb your break?
Yeah.
It's for two things. The first thing is I just wanted to know if it's okay I missed the class in 2 weeks,
Yeah, of course.
and if it's okay for you if I ask you afterwards about things when I go over.
Sure thing, of course.
Okay.
Yeah, sure.
And the other thing is a remark to what you said about the liquor, cerebrospinal fluid, sorry,
Yeah.
I always use the Latin word. As far as I know, white cells are there, but not very many, and red blood cells shouldn't be there.
Should
So
be there.
sometimes there are a few, and one can't really say if it's artificial or not, but they shouldn't be there.
For some reason I remember white and red, but I guess, we have to find, let's see. So, cerebro,
There's reference ranges.
where?
The 4+1, it should say.
Amount,
Yeah, this
reference
one.
ranges.
A bit lower, I think there are 4 other. There's red blood cells, negative.
Yeah, you're right. But wait a minute, where is it again?
RBCs is in the next table. It's this one.
Red blood cells.
None, and
Negative.
the white are almost not there.
Yeah, you're right. So what is it, only an infection, or what do you find it?
Yeah, blood is basically in bleeding, or it's acute.
Getting into the system.
Yeah, and
Okay.
white blood cells, infection, and also you have things like multiple sclerosis.
Yeah.
Different reasons, but in general, cells are quite. It wasn't about. I wanted to be correct.
No, on the contrary. So, but please allow me to make a correction.
Yeah,
For the other students, yeah?
Yeah.
Okay, that's cool.
I just.
Yeah, definitely. Good point.
גם אני
Thank you very much.
default submission שעון יואב. אני גם כאן אתה יכול לבחון. To ask him? אני אמור לעשות אובל, וביקשתי ממנו לדחות את ממש אין בעיה. אוקיי. טוב זה. יכול לך ביולוגיה? של נוער? Yeah, it's pretty good. He's kind of like college level.
מדי, אני מנסה להתמודד עם זה שאני מדבר ליד וזה שאני עושה הרבה הפסקות רטוריות. יותר מדי הפסקות? מרגיש לי פתאום. אז אחרי הזה של RC, בפיזיולוגיה, אז כל הזמן ממש קצת. אני קצת מדי. Enrica is not here, but she has corrected me about a point that should be corrected, the cerebral spinal fluid. So this is the Wikipedia page for it. So it has a certain ionic environment. It has some proteins in it, as we mentioned, and glucose. It does not include any white and red blood cells. In healthy individuals in basal state, it does not include any blood cells. And I said that it has some. So. But I think she's right. If not Wikipedia, then Henrika. So no, really, it's not. Again, with no inflammation, at basal state, should be no blood and white red cells. But hormones you have there. That's the major point from today's class. It carries hormones with it. So let's proceed with the structure and function of the G proteins, and start by saying that G proteins are members in a bigger family of proteins that are GTPases, meaning they hydrolyze GTP. These proteins are tethered or anchored to the plasma membrane via covalent interactions with the lipids in the membrane. And you'll soon see that there is also a monomeric version for the G protein, but in the tetrameric version of it, during intracellular signaling, the alpha subunit dissociates from the beta and gamma, and the alpha subunit is the one that activates the downstream effectors. That can be other receptors or intracellular proteins. We will see it in a minute. But it is most usually the alpha subunit that dissociates from the trimer and activates downstream effectors. And it is the unit that has the GTPase capacity. That's also the unit that hydrolyzes the GTP. And it's not a transmembranal protein. Again, just to emphasize this point, it's a protein that's it is tethered from the inside to the plasma membrane. How do we call these proteins? Proteins that are covalently linked to the plasma membrane but do not cross it. Peripheral protein. So it's a kind of peripheral protein, and it interacts with transmembranal receptor proteins. So in this figure, this protein is the G protein coupled receptor, and that's the trimeric G protein. Okay, and the G protein has several associated proteins that regulate its function in many ways. So, for instance, the rate of GTP hydrolysis that happens on the alpha subunit is enhanced, or at least it is regulated by another protein that is called GTPase accelerating protein. That's a protein that determines how fast, once a GTP have been hydrolyzed to GDP, so in order to activate the alpha subunit again, you have to exchange it, the GDP, with a new GTP. You have to take out the GDP and put a new GTP from the cell cytosol. And the protein that regulates this process on the G protein is the GTPase accelerating protein, short for GAP. Okay, and its activity also is regulated by other factors. Yes, so the GTP, the alpha subunit in it, hydrolyzes a GTP that is linked here in its binding site to GDP. Yeah, guanosine triphosphate to guanosine diphosphate. And after that happens, in order to activate again the alpha subunit, you have to exchange the old hydrolyzed GDP with the new full phosphorus capacity GTP. So you take out the GDP and put in the GTP. And there is an additional protein outside of this figure that regulates this process, and that's GAP. And that's GAP. And there's another one that activates the GTP. No, sorry, GAP is not, no. So GAP regulates how fast the GTP will be hydrolyzed. And the one that determines how fast the GDP will be exchanged with the GTP is GEF, is what we call the guanosine nucleotide exchange factor. Okay, so sorry, my mistake. Again, GEF is the one that regulates the nucleotide exchange process that happens on the G-alpha. And GAP is the one that determines how fast the GTP will be hydrolyzed. Okay, so as I mentioned, there's a monomeric version for G proteins and a trimeric one. So the trimeric G proteins, the second one, the GEF. So as its name implies, it is a guanosine nucleotide exchange factor. It determines how fast the nucleotide will be exchanged in its binding site on G-alpha. Okay? Ask.
Because it changes 3 times.
So, no. So the final time, I'll say it again. So the GAP determines how fast the GTP will be hydrolyzed in the G-alpha, and the GEF will determine how fast a new nucleotide will exchange the old one. That's okay?
Are these 2 kinds of G proteins?
No, these are 2 kinds of regulatory G-associated proteins. These are not G proteins themselves. So the trimeric version of the G protein is directly activated by certain membranal receptors. And the monomeric version of G protein, and these proteins are sometimes called the RAS family because they are all homologous to a protein named RAS. And they are also homologous to the alpha subunit of the trimeric version of G protein. So again, the monomeric G protein is actually only the alpha domain from the trimeric one. And they are also called RAS family. And they are indirectly linked, or they indirectly interact with membranal receptors. They interact with them via additional steps. And these 2 classes of GTP binding proteins, the trimeric and the monomeric one, are regulated in very different ways. And that's just a principle. We will not see how that is. Okay, binding of a GTP to the alpha subunit of the G protein changes its conformation in the following way. There are 2 domains called switch 1 and switch 2, which upon nucleotide exchange, which upon GTP binding to its binding site, interact with the phosphate group of the GTP. Yes, so here you're looking at the GTP because you have the GDP. The scissors are about to cut the last phosphate, but still it's a GTP. And these 2 switches, these 2 domains interact with this phosphate, so they are contracted. And this is the conformational on state of the protein, or of the domain, the alpha domain, of the alpha unit. So in this conformational state, the alpha unit is active, and it activates downstream effectors. Upon GTP hydrolysis, this interaction breaks down, and the 2 switches become loose, and the protein switch to an off state. And in this conformational state, the alpha subunit does not affect any downstream effectors, cannot activate any additional proteins. Okay, so as I mentioned, the most common case is where the G alpha, the alpha subunit, activates downstream effectors. In some cases, it can inhibit them, and in even more rare cases, the beta and gamma subunits are the ones that affect the downstream effectors. So almost all possibilities all options are possible, but the most common process is where the alpha subunit activates the downstream effectors. And what we call a G protein is actually, the entity that we call a G protein is actually different sets of combinations of the units it is built from. Because in the human genome, there are 27 different genes that code for the G alpha alone, 5 genes that code for the beta subunit, and 13 genes that code for the gamma subunit. And a specific G protein is a specific combination of each of these subunits. And this table gives you just the possibilities for the alpha subunit. Again, just to show that you have several ones, and these are, some G proteins, each one has its own specific name, plays a part in a specific pathway via a specific secondary messenger, activates specific effector, and so on. But again, the thing to remember is that the G protein is a combination of different versions of the subunits it is built from.
But
and
it always has the alpha, beta, and gamma?
The trimeric version also always have the alpha, beta, gamma, but you also have the RAS family, which is built only from the alpha subunit. Okay. And each combination has different features. Each one activates in different rates and activate different downstream effectors and so on. Okay, and now about the sequence of events after that are around the activation of the G protein. So everything starts with a ligand binding to its receptor, in this case a hormone, which, the rectangle is frozen. Okay, never mind. So again, the ligand binds to its receptor, activates the receptor, which in its turn activates a membrane-tethered G protein. Once the G protein is activated, the G alpha most usually dissociates from the beta gamma subunits. This complex breaks down, and the alpha subunit flow or move laterally on the plasma membrane because it's covalently linked to it. The alpha subunit itself has a covalent bond with the membrane right here, and it dissociates across the membrane, and by doing that, it encounters downstream effectors. So here you see it activated as some effector, and it most usually, before it will lose its function, it will activate more than one effector. It will activate several effectors. And once the GTP is hydrolyzed to a GDP, the alpha subunit is no longer functional, and it again dissociates across the plasma membrane and meets with its colleagues, G, beta, and gamma, and the complex of the G protein is restored, and the basal state is restored. So just again, the principle about the GTP and the GDP across this sequence of events. So in the basal state, the G protein holds a GDP in its binding site. But resting state, when there is no signal transduction. So when there's no signal transduction, the G protein holds a GDP in it. Upon activation by the receptor, the GDP, it's not that there's another phosphate that's added on this nucleotide, but rather that this nucleotide exits the binding site and a new one with 3 phosphates enters it. And in this case, the G protein is activated. And until it is hydrolyzed, it activates downstream effectors. Once it is hydrolyzed, it becomes unfunctional again.
But it can, the new GTP can come in only after it binds to the receptor?
Yeah, of course. So the binding of the receptor, this activation that happens here, changes conformationally something in the G protein that causes it to have a low affinity for the GDP, so it exits the site. And now the entry of the GTP is actually simply due to the fact that you have high levels of cyclic GTP, the high levels of GTP in the cytosol. So each GDP here that exits the site will be very rapidly phosphorylated once more to become a GTP, and that's something that happens due to an enzyme that is outside this figure, but that plays here in the background. So GDP will be synthesized to a GTP very, very rapidly.
Theoretically, there's no chance that the
Right.
alpha would bind again to GTP?
Exactly. So theoretically, there's a low chance the GDP will enter again the site. But again, it has lower affinity, I think, compared with the GTP to the G alpha.
The activation is when the GDP leaves?
Yeah, the GDP leaves the site, and a new GTP enters it. I wanted to show you a movie that shows this sequence of events, in one shot, but it doesn't work. I don't have the volume for some reason. You can enter the website for the textbook. Just enter the title of the textbook, and make sure you enter the 5th edition at the end. And the first entry in Google will be the website for the textbook. And you have, for each chapter, you have several additional material, and including movies. So if you're interested, the movie is there. Okay, let's mention a method that is used to actually specifically to measure the distance between two molecular components, the distance between two molecules, and in our case, it's a measure of how high did we succeeded in activating the G protein. Because upon activation of the G protein, the alpha subunit dissociates from the beta and gamma. So we will measure the distance between the beta and gamma and the alpha subunit. And that will give us an estimation of how strong did the G alpha have been activated. So it goes like this. By the way, it's called FRET, which stands for Forster Resonance Energy Transfer, and not, as most people think, fluorescence resonance energy transfer. It's not. It's however you pronounce this name, but it's the name of someone. So, okay, so you put an artificial dye on one substance, say the alpha subunit, the blue dye here, and you put in the yellow dye, the second dye, on the G beta gamma. Now you excite the first dye, and when these two molecules are nearby, some energy will be transferred from the first dye to the second dye, which will emit in a specific wavelength. And we can read at this wavelength. And when these two molecules are far apart, then no energy transfer will occur, and you will have zero intensity of emission from the yellow dye. So again, when the two are close by, the yellow dye has high emission, and when they are far apart, the yellow dye has low emission. And so if you put the emission of the yellow dye on the y-axis as a function of time, and you expose the system to a ligand, say the cyclic GMP here, cyclic AMP here, so you see that in some conditions, say in condition B, the GP activation, the G protein activation is quite rapid. You see a decrease in the intensity of the emission of the yellow dye. The decrease happens quite rapidly, really immediately. And in other conditions, it takes several seconds until you get this fall, this drop in the intensity. So that was Forster resonance energy transfer technique. Okay, now let's move on. So that was some principles about signal transduction and about the structure and function of G proteins. Now some of them act through adenylyl cyclase. It's a very canonical pathway. So let's meet the most canonical example for a pathway that goes through adenylyl cyclase. And already now I'll say adenylyl cyclase, as its name denotes, so it's the ones that make a cyclic species out of adenylin or the adenosine nucleotide. Adenylyl cyclase. It cyclates, if you can say so, it cyclates the adenylyl. Okay, so the common example for G protein coupled receptors that act through adenylyl cyclase is for the epinephrine. It's a neurotransmitter and it's a hormone, and it's maybe more common, more famously named adrenaline. So that's the same molecule. And it mediates the body response to stress, mental and physical stress, through various opposing functions or inductions. So it is secreted from a region in the medulla, and it reaches certain tissues of our body, and in each tissue it meets a different receptor and thereby induces different effects. So if epinephrine, if adrenaline will, in adipose tissues, so adipose tissue includes, it's sort of a storage for fatty acids and polysaccharides for time of need, if you need, available energy. So when it meets a specific receptor on adipose tissue, it causes catabolism of glucose and fatty acids. Some glucose and fatty acids are synthesized from this tissue, and they can then reach other tissues of the body. And as you know, these are the reactants for production of ATP. And this happens through the beta-adrenergic receptor, a receptor that is called the beta-adrenergic. When epinephrine meets certain receptor on certain cells in the heart, so through beta-adrenergic receptors, it will cause increase of heart rate. And now, increased heart rate will cause more blood flow to the tissues, right? And most usually to the muscles. But in parallel to this, you have a cutting off of circulation of the blood flow to some unneeded organs, such as the skin. Some people say that this is why we become pale while we are scared and so on. So unneeded organs, we have skin, digestion, and I guess some other organs. And this induction happens through the alpha-adrenergic receptor. Maybe even a more impressive example for what epinephrine can do. So it can act on the same tissue, for instance, smooth muscle. And the smooth muscles around respiratory tube, upon exposure to epinephrine, will loosen, and so to enable more respiration to pass through this wider tube. And the same tissue, smooth muscle around arteries, upon exposure to epinephrine, will tighten to increase the blood flow, to increase the blood pressure. And again, these opposing effects, as you see here, or in the last example I gave, are thanks to different receptors that are localized in each tissue. Yes?
Can you repeat what the
Adenylyl cyclase is a receptor, is a downstream effector. It's a protein that produces this species of adenosine monophosphate. It takes an adenosine monophosphate and it cyclates it. It makes it Can I say it cyclates it? The native speakers, well, they don't just go out. So I guess I can say it. So it makes cyclic AMP. And all I've described so far are the different receptors that the epinephrine meets in the target tissue. So in a minute we'll get to the adenylyl cyclase. But again, the first thing to know is that each hormone, and in our case epinephrine, meets different receptors, and sometimes it induces different reactions simply due to the different receptors it meets.
But in all of these three different cases, it affects the
adenylyl cyclase, yes. So let's see it in the next slide exactly. So the first couple of effects are induced through the beta receptors, which interact. I have to stand. Presentation is so huge. Okay, so when it interacts through beta-adrenergic receptor, they interact with stimulatory G protein. And if it's a stimulatory G protein, it activates the adenylyl cyclase, which cause more cyclic AMP to be synthesized. And the same hormone, while it is recognized by the alpha-adrenergic receptors, which interact with inhibitory G complex, which inhibits adenylyl cyclase and cause less cyclic AMP. So both sides of the effects that I've mentioned go through higher levels or lower levels of cyclic AMP through these mechanisms. The other one, this is the alpha receptor for adrenaline, and this is the beta receptor. Yeah. and the first one inhibits it, while the others activates it. While the other activates it.
Do they exist in the same cell?
No, most usually a cell will express the one or the other, because these will be opposing effects. And yeah. Okay, what about the structure of adenylate? So now we are focusing on the structure, the basic structure of adenylate cyclase right here. It passes the membrane 12 times. The catalytic site of adenylate cyclase is built of two cytosolic domains that are joined together into a globular dimer. Yeah, these two domains joined together into a globular dimer. And this is the site of interaction with the G-alpha that activates this catalytic site upon the adenylate cyclase. Now, as is the case for the NMDA receptor, even to which the crystallography structure was achieved thanks to a substance that holds it in a specific configuration, and other examples for crystallography structures are manifestations of the same principle. Also here, clusterization of these two. Also here, first of all, the protein was not crystallized as a full protein, but only the two catalytic subunits were crystallized. And it was enabled thanks to a substance called forskolin that sits right here and glue these two subunits together, which enabled researchers to clusterize to make a crystal out of it, and then get its specific structure. And now the interaction with the G-alpha is not based on a crystal. It's based on the amphipathic and hydrophobic nature of specific regions along the G protein, and we think that it is the switch 2 domain that interacts with the catalytic domain or the catalytic site of adenylate cyclase, and another region that's called alpha 3 beta 5. So that's how the complex of a G-alpha and the catalytic site of adenylate cyclase look like. Now, virtually all the diverse effects of cyclic AMP highering of its levels or lowering of its levels go through the function of a protein called protein kinase A. And it happens with the following mechanism. So the cyclic AMP binds to two sites on each regulatory unit of the pKa. So the basic structure of the pKa is that it's built out of two regulatory subunits that hold two catalytic subunits. And upon cooperative binding of the cyclic AMP to one site that induces higher affinity to the second binding and to the third and fourth binding, and while all sites are populated will be more. So while all sites are populated with cyclic AMP, the catalytic sites are released from the regulatory. The catalytic units are released from the regulatory units, and then can be, are active and can induce specific function. In our case, phosphorylation of other target proteins. When you have no cyclic AMP here, the catalytic unit sits here because this region in the regulatory unit just very much resembles the target sequence on a target protein that's supposed to be phosphorylated. It looks like the substrate of the catalytic unit, only it's called a pseudo-substrate. And it's called a pseudo-substrate because it can't be phosphorylated, and that's the trick. So once the catalytic unit tries to phosphorylate this region, it's just stuck here. And upon cyclic AMP binding, there is some conformational change that releases these catalytic units. So the phrasing the researchers sometimes use is the following. Let's see, we understand it because we learned the terms that are in it. So binding of the first cyclic AMP to pKa lowers the dissociation constant for binding of the second one. Why is it a positive cooperativity that's described here and not a negative one? So remember, the dissociation constant is how hard the two molecules, how fast will they dissociate. And if we talk about lowering of the dissociation constant, it's like increasing the binding constant, if there was any. So it's increasing the binding of the second cyclic AMP. So this one increasing the binding of this one, which increases the binding of the third and the fourth one, and so on. So this is, good example of a positive cooperativity. Yes? So phosphorylation is downstream, so we will get to it in a minute, but these catalytic units, phosphorylate target proteins. Add phosphate groups to other proteins that are not yet.
You explained why you have the catalytic site and
Why do they stick on the regulatory units? So because these areas in the regulatory units look like a protein that needs to be phosphorylated by the catalytic unit. It has the exact same structure, the exact same sequence, only it can't be phosphorylated. And so when the catalytic unit tries to phosphorylate it, it stays right here. On a typical cell, we have several thousands of membranal receptors. And still, upon activation of a given hormone, by a given hormone, there is an increase of several millions, or more than that, of cyclic AMP molecules inside the cell. So there is some amplification between these two steps, right? There has to be some amplification. And more specifically, the first amplification happens due to the diffusion of the receptor and the G-alpha from the G protein across the plasma membrane. Because remember, we said that once the G-alpha is activated, it travels around the membrane, encounters several downstream effectors, and activates them. Not just one, but several ones. So there's an amplification simply due to the diffusion or simply due to the plasmatic nature of the membrane. So again, you have to have certain fluidity in the membrane, and we've discussed this issue, in order to get enough amplification of GPCR pathway. So that's one of the functions that the plasmatic membrane is important in. Okay. Now, between the step, once the cyclic AMP levels have risen, so this transition between the cyclic AMP and the activation of the protein kinase A does not really have any amplifications, because you have to have the cyclic AMP molecules bind to the pKa to keep it functional. They have to stick with it as long as it's functional, so there's no real amplification right here. But then each protein kinase A will phosphorylate not just one, but many downstream target proteins or enzymes. And each enzyme, as you know, produces not just one product, but many downstream products. So there's also amplification in the downstream transitions. And roughly speaking, the more steps in such a cascade, the greater the amplification, but the slower the process. And the most substantial example for such amplification will be given in vision, which we will learn, I think, just next time. Where a single photon that's absorbed in our photoreceptors can still make us be aware, cognitively aware, to it in space. There's some experiments to show that it's real, this result, and it's due to a very robust amplification that happens in the retina. Okay, some factors that contribute to termination in the case of adenylate cyclase pathway. So first, the affinity of the receptor for the hormone, once it is activated, reduces after it is activated and activates a downstream G protein. Again, after the receptor is activated and activates downstream G protein, its affinity to the hormone extracellular signaling molecule reduces substantially.
And how is it increased again?
It's not increased, but these are the factors that terminate the signal, terminate the response. So if you still have the hormone outside the cell, it will be less effective in transducing the signal, simply because of the affinity between the receptor and the hormone. Okay, so the following features are the ones that are responsible for the termination of the response to a hormone, in the case of the beta-adrenergic receptors.
How does it start again? And what starts the
So the less
Again,
yeah.
what is the affinity?
So there's some kind of, so the lower affinity between the receptor and the hormone is due to some, as you got a picture by now, to some conformational change, and there is some kinetics to it. And once the receptor is it is restored its basal state at a certain point, either that or there have to be a new receptor that to be synthesized and localized on the plasma membrane. Okay, one of the downstream effectors of PKA, not of PKA, sorry, of the G protein is an enzyme called cyclic AMP phosphodiesterase. So remember, one of the products or the steps of the G protein through beta adrenergic receptor pathway increases the cyclic AMP, but another enzyme is initiated or activated in this process, and its activation maybe happens with a certain delay, not to abolish any signal through this pathway, but it becomes functional. It's a cyclic AMP phosphodiesterase that lowers the level of cyclic AMP. So it's sort of like the sodium channels in action potentials that allow depolarization, and with a certain delay, with slower kinetics, the potassium channels that allows restoration of the hyperpolarization. So this process happens with a certain delay, with much longer time constants, obviously, than action potentials, but still it restores, very actively restores the basal state of the cell. So that's the second feature that allows termination of the cellular response. Now, upon prolonged activation, or tetanic activation of the hormone on its receptor, so additional things happen, and usually you have phosphorylation of several residues on the receptor itself, the receptor on the plasma membrane that have received the signal. Now, if we sometimes have phosphorylation on the same receptor that has received the signal, and we sometimes have phosphorylation of more than just the receptor that has received the signal, but on other receptors as well. So first of all, this is the plasma membrane. Here's the receptor that received the hormone, the beta-adrenergic receptor. Here's the hormone. If it is activated prolonged a long duration of time, then some processes are initiated in which so some things happen here, several steps, and some of them activates kinases that phosphorylates. These will be kinases that phosphorylates either just the receptor that activates that initiated the whole cascade, in which case it will be called a homologous desensitization, or they can phosphorylate this receptor and also additional other receptors, in which case it will be called heterologous desensitization. So these are different receptors. Let's plot them like that. Okay. Bark. So there is a specific example to homologous desensitization on the beta-adrenergic receptor, and it's caused by an enzyme, a receptor-specific enzyme that's called beta-adrenergic receptor kinase, or simply BARK. And this one desensitizes only the receptor that initiated the cascade. So BARK performs homologous desensitization, in opposed to other kinases which you've not mentioned their names, which induce also heterologous desensitization. So in homologous desensitization, the number of phosphates per receptor can be roughly used to estimate how much ligand has been bound in the recent past, say 10 minutes back. Because again, if you expose this hormone long enough to the receptor, will initiate a cascade in which some kinases will, if you have a pure homologous desensitization, then these receptors will start to accumulate phosphate groups upon it. These are phosphate groups. And if you have a way to measure how many phosphate groups are linked to this receptor, and you know that you have a pure homologous desensitization happen here, so you can measure how many, or at least compare between two states, because this will be proportional to the amount of hormone that was present in the system. Okay, and now if the beta-adrenergic receptor is phosphorylated enough, there's another protein, cytosolic protein, that joins the party. It is called beta-arrestin. It recognizes extensively phosphorylated receptors, beta-adrenergic receptors, and while it joins the party, it completely shuts off any further signaling through this receptor. So again, this is the G protein coupled receptor, which was activated by beta-adrenergic receptor. And once this is the beta-adrenergic receptor as I described in this plot. And while it is sufficiently phosphorylated, a protein named beta-arrestin, a cytosolic protein, recognize this phosphorylation area, binds to it, and shuts any further activity through this receptor. What is its activity? It activates downstream G proteins. So once the G protein coupled receptor is phosphorylated enough to be recognized by beta-arrestin, it's able to activate any downstream G proteins. And this beta-arrestin, this protein is a good example of it's maybe the best biological manifestation to the transition between a short-lasting effect in GPCR pathways to long-lasting effect. Because while it shuts off the short-lasting effect, the G protein activation, it initiates, it's a starting point for other additional processes, intracellular processes, that has to do with transcription of new genes. Because beta-arrestin interacts with several proteins that initiate different processes that eventually would produce transcription and translation of new proteins. So again, you see that it stops the short-term effect, the direct specific activation of G protein, and it transfers us to the long-lasting effects, which are, among others, transcription and translation of new proteins. So this is how beta-arrestin is the linking chain. Yeah, the linking chain between these two domains that we distinguish the short and long-term effects. Okay, there's a spatial localization of most of the players that in this pathway, the beta-adrenergic pathway. So, and that's because a rise in cyclic AMP in some places of the cell can be good, can be beneficial, but in others can be deleterious, and that's why we need to sort of spatially localize the components of this pathway to make it spatially focused or spatially localized. And now I'll show you how spatial limitation determines temporal limitation as well in the case of the beta-adrenergic pathway. So as we said, upon cyclic AMP elevation in the cyclic AMP levels, a protein called pKa will be activated. Now I tell you that this protein interacts with what we call pKa-associated proteins. It's just a complex build of several resemble proteins that links it with another protein, phosphodiesterase. That's the cyclic AMP phosphodiesterase. That's the protein that breaks down the cyclic AMP and brings us back to the linear form of AMP. So upon cyclic AMP increase, pKa will be activated. What do we mean by that? We mean that, when the regulatory units are populated fully with the cyclic AMP, the catalytic subunits are released, and they phosphorylate other targets, among them PDE. Now, when PDE is phosphorylated, it is activated, but what PDE does is in contrast to what the initial catalysator was, because it reduces the level of cyclic AMP, which will cause dissociation of cyclic AMP from the regulatory units of pKa, and will cause the catalytic units to come back to their home base and to the basal resting state to be restored. Okay, so we started with increased levels of cyclic AMP, and we ended intrinsically in the same pathway with a negative feedback that brings us back to low levels of cyclic AMP. Yeah. So if you put these two proteins in the same vicinity, any increase in cyclic AMP levels will not be only limited to this region in the cell, but will also be temporally limited quite effectively, because so again, the PD will meet the cyclic AMP, will feel the activation through the phosphorylation by pKa, and will direct, just because it will happen very fast. And it happens fast because you put these two proteins in close proximity, and then it happens very fast, so you have a limitation, a temporal limitation or temporal restriction of this process.
So the cyclic site wouldn't move farther away because of its mechanism.
The cyclic AMP will not be distributed far away.
But why? Where does the cyclic AMP came from? It can come from a long distance as far as this mechanism.
Yeah, as far as this mechanism, okay. Well, I guess maybe some other components are stick to the same complex. I don't know. But all I know is that the pKa through pKa-associated proteins is linked with PDE. You're right. Maybe cyclic AMP will increase in certain levels in the cell. Well, I guess the levels of cyclic AMP increase is also localized in some process. I'm not sure. What we do know is that this process plays a role. Okay, and for instance, what we've shown here are localization of protein kinase A in nuclear membrane in heart muscles. I think by now that people have found the same mechanism in other tissues as well. Okay, so these were G protein kappa receptors and the major pathways that affect adenylyl cyclase, that goes through adenylyl cyclase. I think I won't start the big point about the ion channel, but just as a teaser to next time. So again, we will see here the molecular basic for vision at the level of the retina, and only for rod cells. For those of you who are already familiar with rod cells and cone cells, we will describe the molecular basis for the rod cells in the retina, because it passes through G protein kappa receptors and it regulates ion channels in it. Yeah, so let's stop here, and I'll see you next time.